Today we're going to be diving into our DNA topic a little bit further and looking at a mutations PowerPoint and worksheet. By now you should have read through the mutations worksheet and PowerPoint, and you may be confused on what to do next. So this chart below is asking us to transcribe the DNA sequence into messenger RNA, then use the codon chart or wheel, and I have one below, to indicate what amino acids are being coded for by the base sequences, and then decide what type of mutation is illustrated. Our choices are going to be a point mutation and a frame shift. A point mutation changes what amino acid could be produced, or it may not, and that's a change in one letter, it does not change the length. And a frame shift mutation makes the DNA sequence longer or shorter by inserting in there a new nucleotide, and that can change the entire protein produced. So just like you did on your homework from last class, you are going to take DNA and turn it into messenger RNA. And so for this one, I'm going to zoom in. TAC becomes AUG, then it becomes CGG, UCA, CCA. And then we're going to go down to our wheel, and on our wheel, we're going to see what AUG codes for. AUG codes for methionine. So then we're going to go back to our chart and write MET, which is abbreviation for methionine. Then we're going to go back CGG, which is arginine, ARG. UCA, start in the center, UCA becomes serine. And CCA, our very last one, CCA is proline. So for those of you that were struggling with that, make sure you start in the center and then go out. So now that's our original DNA sequence, and those 4 amino acids will code for a certain protein that does a certain job. And so now we're going to see what our mutation is. So same thing we did in the example. We're going to convert our DNA into messenger RNA, and we're going to do that by writing AUG, GGG, UCA, CCA. And so AUG we know is our start codon methionine. GGG though is different, so we need to go back to our chart. And GGG is going to give us glycine. GLY, abbreviation. We found UCA previously as serine and CCA as proline. And so we can tell that we have a different amino acid here. So that is going to be a point mutation. It does not change the length. This is specifically called a missense because it will change the amino acid, therefore the protein produced. And the very last one we're going to do, we're going to look at another one. So we have TAC, which we know is our start codon, and that is AUG. Then we have GGU. Then we have CAC. Then we have a random CA at the end. So AUG is our start codon, that's methionine. We have to go down to our chart, and GGU is glycine. Then we have CAC, which is histidine. And you have to have 3 nucleotides to make a codon. So this random CA right here can't make an amino acid. And what you'll notice is that our amino acid sequence is different than in the original. And you will also notice that in this, we also have a different length. And so we're going to call this a frame shift. And so a frame shift will make our DNA sequence longer or shorter. So what you need to do on this worksheet is use the PowerPoint to answer the following questions. And then you have another example on the third page of how we can go from DNA to messenger RNA to a chain of amino acids, which is a protein, and how it can be changed by mutations. Mutations are not always bad. Keep that in mind. Mutations allow living things to grow, adapt, and evolve over time, but sometimes the mutation in the same area can lead to a genetic anomaly or a bad mutation such as cancer. If you have any questions, please feel free to reach out to me via email. And I hope that you guys understood everything.